Good morning. Thank you very much, Cecile, for that wonderful introduction, and thank you to all of you for coming here to listen to my talk so early on a Sunday morning. Why is it that a lineage that has been diversifying successfully through evolutionary time has a branch that suddenly begins to diversify massively to drastically change the environment it lives in? This is what happened with angiosperms, and this is one of the reasons why I find them to be completely fascinating. Angiosperms are one of the most species-rich groups among metazoans. They include an extraordinary morphological diversity, and much of this morphological diversity is based on innovations that represent modifications of structures and functions that are present in other lineages of seed plants. However, they include, for example, completely new ways of building trees. They include the complete redefinition of the herb habit. They have built new types of cells in their vascular system. They had the great idea of joining male and female reproductive organs in a single functional unit, which is the flower. And the flower is a hub of innovations. They include the carpel and a second layer surrounding the ovules. And within the embryo, they have a triploid nurturing tissue that is formed only after there is fertilization. Also, flowering plants have built a new set of complex interactions that did not exist before flowering plants came about. Very importantly, they represent the keystone of modern terrestrial ecosystems by providing the structural and energetic basis of most extant biomes. However, there are many things that we don't understand about angiosperms. For example, we don't really understand who are their closest relatives because phylogenetic relationships are not well understood. Also, as I said, there are many innovations, but we're not very clear about what is their homology among other plants. Also, flowering plants are a classic example of the rock versus clock conflict, in which ages estimated with molecular clocks, of course based on molecular data, provide substantially older ages than the first appearance of the group in the fossil record here, or of many orders which are here shown only in the Cretaceous. So trying to investigate the timing of angiosperm diversification has been a major topic in my career in the past few years, and I would like to talk a little bit about this in this presentation. An example of the work that has been conducted is a study that was published in 2015 with my colleagues, in which we included representation for approximately 90% of the angiosperm families. And this tree was calibrated with 136 fossils. In addition, we included a constraint on the age of the crown node derived from paleobiological methods. We are expanding now this work in collaboration with my colleagues Santiago Ramírez Barahona and Hervé Soquet. And in this new approach, in addition to increasing the molecular data set, Santiago really has done an outstanding job at increasing the taxonomic sample. He has included over 1,200 extant species, but very importantly, these extant species represent 100% of the families. And what is very relevant is that these families are being sampled by their crown node. As you probably remember, a crown node is defined as the least inclusive monophyletic group that includes all the living species of one particular clade. And in order to sample the crown node, it is necessary to include at least 2 species. But these 2 species cannot be sampled randomly. It is necessary for us to have a knowledge of the phylogenetic relationships within the clade. For example, in this family, which is the Cyparininaceae. And we need to understand its phylogenetic relationships, because we need to know which are the two branches that stem from the crown node. And it's necessary to sample one species from each of the branches stemming from the crown node. So in this example, it would be necessary to sample this species and any one of the species belonging to this clade. So Santiago very carefully went through the angiosperm phylogeny website, downloading the most recent phylogenies for families, and he was able to sample the crown node of 85% of them. For the other ones, either no phylogenetic information was available or they represent monotypic families. So this sampling is really extraordinary, and in addition to the node dating analyses that Santiago is conducting, we decided also to estimate the timing of diversification of the angiosperms using the fossilized birth-death process. We are collaborating in this project, in addition to Santiago and Hervé, with my student, Rebecca Hernandez, and with the FBD team that include Tanya Stadler, Tracy Heath, and Joel Barrido-Sotani. The fossilized birth-death process is a method that includes living and extant species in the same evolutionary process. Fossils can appear as terminals in extinct lineages or as ancestors in lineages that are present in the present day or that went extinct after the fossil was sampled. In addition to estimating divergence times, the fossilized birth-death process also estimates the rates of speciation, extinction, the probability of fossil recovery, and the probability of sampling an extant taxon. And it can be done in such a way that incomplete and biased sampling of extant taxa is accounted for. In this presentation, I will be talking about the angiosperm fossilized birth-death process, but I will be focusing very much on the problems that I encountered when sampling fossils. First of all, we sampled fossils within the crown group, and this was a large work done by Santiago and by Hervé. Basically, what we did, the angiosperm fossil record is of relatively good quality. Many of the clades are represented, but most of them are known from a single locality and from one or very few specimens. In our case, we used as basic data an updated database that was prepared in my lab several years ago that includes about 3,500 records. From these, we selected the fossil that represents the oldest fossil for particular clades, which were, in this case, families or subfamilies, and we very strictly looked at primary references. We also followed best practices by recording the name, the holotype, the detailed locality, revised stratigraphy, the updated age in case it was relevant, and the primary reference. And all this was recorded in the dataset Proteus, which was created and is maintained by Hervé. Although in the FBD it is possible to include morphological data to place the fossils with respect to living taxa, in our case we did not have morphological data. So we placed the fossils manually among the extant lineages. This is not ideal. It has a number of drawbacks which are shared with no dating. For example, because of morphological convergence, if we base those assignments only considering general aspect or a suite of characters, the fossils can be placed erroneously. It is slightly better if among the attributes of the fossil, there are some that are apomorphies for particular clades. But nevertheless, that can also be problematic because in some cases, the parts of the fossil that are not preserved might show completely different features. And also in the case of angiosperms, we don't really know which are the apomorphies of all of the clades. Ideally, it would be the best to try to place the fossils directly based on explicit phylogenetic analysis, based either on morphological data or on total evidence. But even this is not mistake-proof. And as an example of that, I would like to tell you about this beautiful fossil flower that is three-dimensionally preserved. This flower is from the late Cretaceous, from the Turonian of New Jersey. And the authors who were studying it, first based on its general characters, considered that it belonged within the order Saxifragales. And then they went ahead and collected morphological data for a number of placeholders within the Saxifragales and outgroups, and they also obtained molecular data and conducted a parsimony total evidence analysis. According to this analysis, the two species of this fossil, which is called Diversistylus, fell very closely to family Itaceae. Several years later, my student, Andrea Lopez Martinez, in her master's thesis, she was interested in looking at the evolution of floral structure at the early diversification of eudicots. So in this clade, this is part of Andrea's tree, and she included placeholders for outgroups of the large clade pentapetalae, all belonging to the eudicots. She had a very complete morphological data set for extant species, which are here shown in black, and for fossil species, which are in red. In her data set, she included the Saxifragales and Diversistylus, and according to a total evidence Bayesian analysis, Diversistylus fell within the Cornales, so it did not fall within the Saxifragales. And Cornales belongs to the superasterids, and Saxifragales belong to the superrosids, which represent major divergent clades within the pentapetalae. So this is an example in which if the fossil is initially placed into a clade where it doesn't belong, then there is no way that phylogenetic analysis will place it correctly. So those were some of the issues that we faced while assigning the position of crown node fossils. But I think that perhaps some of you are aware of these complications, but what was entirely new, at least to me, were the complications that would be associated with trying to identify and to assign stem lineage fossils. It has been shown in several studies that including stem lineage fossils is very important for fossilized birth-death analyses. In the case of angiosperms, this became complicated, and I would like to go into some of the details of the issues that I found. Some of them are only relevant to angiosperms, others are relevant to seed plants, and others are relevant to general fossils, plants or animals. The first thing that I encountered is that angiosperms represent an evolutionary singularity. By this, it is meant that along their evolutionary lineage, many things happened that resulted in very deep modifications in their phenotype, in their genotype, and these modifications are not replicated elsewhere in the tree of life. In particular, some of these represent apomorphies, including specifically the carpel and a structure which is called the second integument of the ovule, for which we really don't understand their homology with respect to other seed plants. And I will be talking a little bit more about those two later in the talk. Another issue that is relevant to seed plants is that while there are only 5 living lineages, which are cycads, ginkgo, nitales, conifers, and angiosperms, the potential list of extinct lineages is very large. This is a list taken from a paleobotany textbook, updated with information from new publications, but we don't know if these groups are monophyletic. It seems now that at least some of the names that are here actually correspond to the same lineage. And also, it's entirely possible that within each of these groups, there are 2 or more divergent evolutionary lineages that need to be separated. So we really have very little information about these extinct lineages. Another issue is that we have really a limited understanding of phylogenetic relationships among angiosperms and other seed plants, and this is a bit of an understatement. For example, here I'm showing examples of phylogenetic trees estimated with morphological data for living and extinct seed plants. The branches in color are living lineages, and the ones in black are extinct. Angiosperms are always in red, and one of the things that you can see here is that among living lineages, angiosperms and a group which is called Nitales, which are here shown in blue, always come together as most closely related among living lineages. In some cases, there are some fossils that come in between them, but among the living taxa, angiosperms and Nitales are most closely related. The results of molecular phylogenies, when they started to be available about 20 years ago, initially were very unstable, but more recently they have become more stable, and they indicate that angiosperms are the sister taxon to all other living seed plants, and that Nitales are more closely related to the conifers, either a sister group of them or embedded within them. So the single result, well, not the single, but the result that seems to be most consistently coming out from molecular phylogenies is that angiosperms and Nitales are not closely related. And this is the single result that consistently seemed to be coming out from morphological phylogenies. So there is a significant conflict, and the realization of this conflict caused very substantial distress some 20 years ago. Another issue that is relevant to plants, but also to organisms that are not preserved in whole, is that different parts of the organism are preserved separately. For example, these seed-bearing organs were described by Fabiani Herrera and his collaborators. This material is preserved as three-dimensional lignified mesofossil, so it can be studied with scanning electron microscopy. These seed-bearing organs were found in central Mongolia in the early Cretaceous. In the same sediments, they found these leaves that have parallel veins and resin canals, and after detailed examination, Herrera and collaborators found that both in the seed-bearing organs and in the leaves, there are these very distinctive stomatal apparatus with enlarged guard cells and a very distinctive organization of the subsidiary cells. And also, both in the seed-bearing organs and in twigs that also bore the leaves, they found structures bearing these very distinctive unicellular trichomes. And it was based on this type of morphological evidence that they were able to realize that the leaves and the seed-bearing organs belong to the same plant, and they were able to make this very nice reconstruction. But please be aware that the seed-bearing organs were assigned to genus Umaltolepis, while the leaves were assigned to Pseudotorrelia. So this is a case in which a single species is known in the fossil record by two or more names. The contrary situation is also present in the case when organs that look morphologically similar, even though they belong to different lineages, are assigned to the same form genus. This is the case of Nilsonia, which is a form genus for leaves that belong to different lineages within the cycads. So this is a case in which the same different species are assigned to the same form genus, and I think people should be aware of these kinds of situations, at least in the fossil record of plants, because if data is downloaded from public databases, there should be a realization that the number of names is not directly correlated with the number of species. Another example are conflictive interpretations, even when the material is exquisitely preserved. As an example is the number of layers surrounding the ovules of Benetitales. So the ovules are unfertilized seeds, basically, and Benetitales is a lineage of extinct seed plants that existed from the Triassic to the Cretaceous, possibly even extending to the early Tertiary. They are characterized by cycad-like leaves, but they have a very distinctive type of stomata. They have flower-like reproductive organs, either unisexual, male or female, or in other species they can be bisexual, bearing in the same organ female organs and male organs. And because of this, they have been considered in many phylogenies as potential relatives to the angiosperms. And in this context, understanding the number of layers surrounding the ovule becomes relevant. And the reason why, to explain to you why this is important, I would like to very quickly review the parts that are present in a basic ovule and in an angiosperm ovule. Going from the inside to the outside, there is the embryo sac, which is technically known as the nocellus, which is surrounded both in angiosperms and in the basic ovule by one layer. But then in the angiosperms, there is a second layer, which its homology in other seed plants is unclear. But very importantly, the plesiomorphic condition in the angiosperms, in this second layer, is that involves a curvature that is produced by asymmetric growth. And this is very important because when morphologists are seeking homologies for the second layer among other plants that are not angiosperms, they need to account for this curvature. Another structure that is present in angiosperms is the carpel, and of course the carpel is not part of the ovule. The carpel is external to the ovule, but again we have very limited understanding of the homology of the carpel. And I would like to emphasize that the ovules are born on the carpel. So this might be evident to botanists, but the ovules are not born on the tip of the flower or on the tip of an axis. They're born on the carpel. And it's something that also needs to be taken into account when seeking homologies of this structure among other plants. So going back to the fossils, back to the Bennettitales, Kai Peterson and collaborators in the late 80s they looked into considerable detail to these ovules assigned to genus Vardacleftia from the Upper Triassic of Greenland. And these ovules are among the oldest within the Bennettitales. And they describe them as having two layers. This would be the inner layer, and this one here would be the outer layer. Many years later, Ruth Stocky and Gar Rothwell described material from Vancouver Island in Canada, where the material is permineralized. These are calcareous compressions, and the preservation is absolutely exquisite. For example, here, this represents the embryo. You can see the two cotyledons, and in this section, they very clearly showed that there is a single layer. However, since these Bennettitales are from the youngest part of the range, the question emerged that maybe Bennettitales early in their evolution had two layers, but later one was lost, or maybe that they were different evolutionary lineages within the Bennettitales. More recently, Pott studied material from the Upper Triassic of Austria. This material represents Coley compressions that were macerated. So this author was studying cuticles. These are cuticles. And he was able to conclusively show that there is only one layer associated to the Bennettitalian ovule. He not only studied this material, but he actually conducted a number of comparative analyses with other fossils, also from the earlier part of the range. He was not able to study directly Vardacleftia, so the question still remains, but these studies based on very well-preserved material seem to indicate that possibly the most common condition within the Bennettitales is a single layer, or that Vardacleftia perhaps represents a special situation, but this needs to be examined in greater detail. And of course, knowing the number of layers in these fossils becomes very important because this is a key feature that can be scored in phylogenetic analyses for better understanding phylogenetic relationships among seed plants. Finally, on the bright side, new field work has led to new discoveries, new interpretations, and new data. I have already mentioned to you material from the locality in central Mongolia that Peter Crane and his group have been studying. This is a different type of seed-bearing organ. These are seed-bearing organs that have a stalk, then it divides, and this structure is called the cupule, and inside there is an ovule. These structures have been found to be attached on a lax cone. And this material is three-dimensionally preserved, and it's usually studied with scanning electron microscopy that allows to see many details of its morphological surface. However, more recently, Crane and his group have been studying these fossils with computerized tomography. I can't seem to find the mouse here. Okay, here we go. And the amount of detail that they have been able to see is really impressive. For example, you can see the micropyle here. You can see the point of attachment of these structures. This is a longitudinal section. And they were also able to do a transverse section. This is the stalk. These are the ovules. These are the so-called flaps of the cupules. And the fact that they are being able to do these computerized tomography analysis is providing very large amounts of data that can provide very detailed morphological descriptions, which in turn can be placed in explicit morphological data sets that can be included in phylogenetic analyses. It is not clear if these particular fossils are on the stem lineage of angiosperms, but they're going to have a very important role in better understanding phylogenetic relationships among seed plants. So, going back to the angiosperm fossilized birth-death process, the things that I have just spoken about are the things I ran to when trying to select angiosperm stem lineages to include in the FBD analysis. So first of all, it was very difficult for me to decide which lineages to include. So to do that, I considered the work by Mario Coiro and collaborators who conducted a split network analysis, including different databases for morphological studies for extinct and living seed plants. In these studies, the angiosperms are here, the cycads are here, and this, according to this analysis, these would be their closest living representatives. And between them are a number of lineages which would be the angiosperm stem representatives. These lineages are the Petraliales, Catoniales, Glossopteridales, Pentoxylales, Benetitales, and also I included some angiosperm-like pollen grains from the Triassic. In these cases, some of these groups have a very large fossil record, for example, the Benetitales and the Glossopteridales, but I was very strict trying to include only fossils for which the morphological features had been explicitly included in previous phylogenetic analyses, or for which we had unequivocal anatomical information that linked them with those fossils. So in the end, we ended up with only 49 representatives of angiosperm stem relatives. We conducted two different fossilized birth-death analyses. In the first one, we included only one terminal per family, leading to 433 living species. We included 234 crown fossils, 48 stem fossils. We conducted the analysis sampling for ancestors, and we conducted analysis assuming that sampling is uniform or accounting for incomplete and biased sampling. This is the result of the uniform sampling analysis, in which you can see here the angiosperm stem relatives which do not reach to the present, and this represents the angiosperms with terminals that do not reach the present because they represent the crown group fossils. The age estimated for the angiosperms is 318.9 million years, which places them back in the Carboniferous. We conducted also a diversified sampling analysis in which the fact that sampling of living species is incomplete and biased. And here, the estimate for the age of the crown node of the angiosperms is much younger, is 167 million years, which places them in the middle of the Jurassic. We conducted a second study. This analysis is still very much in progress, in which we included the complete taxon sampling obtained by Santiago, in which we are sampling the crown node of angiosperm families. The representation of fossils is the same, and here you can see the stem lineage fossils in yellow, and in here, the red branches represent the crown fossils interspersed among living species. I will be concentrating on the diversified sampling analyses. First of all, I would like to mention that the Markov chains take a long time to converge. And even when they do, the effective sample sizes of different parameters are still very low. So these chains need to run longer. However, the results are looking very exciting. For example, here we have the stem lineage representatives. The angiosperm crown node is dated at 172 million years, placing them in the middle of the Jurassic. The mesangiospermi, which include about 99% of living angiosperm species, are dated to the upper Jurassic to approximately 156 million years. And the crown nodes of the magnoliids and the monocots are placed in the uppermost Jurassic or at the onset of the Cretaceous. Eudicots are dated as 140 million years in the earliest Cretaceous, and pentapetalae, which include about 70% of living angiosperm species, are dated to about 125 million years in the middle of the early Cretaceous. So these results seem to be very promising. We are very interested in comparing the estimates obtained here of the diversification within angiosperms with the estimates that are being obtained in our node dating study. So to conclude, I think that including fossils is very important for macroevolutionary studies because they provide information about past richness, past form and function. They provide direct information of speciation and extinction, and they can provide different types of calibrations for different types of dating analyses. But including fossils can be problematic because the fossil record is incomplete in different ways, and also because different organisms may be incompletely preserved. So, however, I think that we can obtain information from the natural history of organisms, and we can use that information to better inform parameters in FBD or in other types of macroevolutionary analyses. And I have some explicit ideas about how to go about this, so if any of you would be interested in working on this, please talk to me. Regarding the angiosperm fossil, the age of the angiosperms, we are seeing that the use of more realistic models and of critically analyzed data is providing estimates that are more congruent with independent evidence, specifically with the evolution of morphology, with the timing of appearance of lineages in the fossil record, and also with increasing diversity and abundance in specific localities. In congruence with the analysis that has been being done by Santiago based on node dating, we are finding that many of the angiosperm families became differentiated in the Cretaceous, but began to diversify into extant richness in the Tertiary. And the bottom line, I think the take-home message from this presentation is that while including fossils in macroevolutionary studies is very important, it is also very important that they are accurately placed in terms of the phylogeny and also that their age is correct. So I would like to remind you that using correct data is equally important as using more realistic models. And I think that an avenue towards this goal is joining forces between organismic biologists, and people who develop models towards better understanding of macroevolution. And I would just like to finish by thanking my working group, my students Adriana, Rebecca, Andrea, and my colleagues Santiago and Hervé. And I would also like to thank the FBD team, Tanya, Tracy, and Joel. I would like to thank logistics support and funding from my university. And very specially, thank you very much for your attention.